Hello, Science Streams, welcome. You don't have a shout out. I need to fix that. For people who I know who stream, I usually have something more automatic so no one has to type anything in. I need to write that note down. If I had a pen, I'll remember. I'll remember. Eventually, at least. All right. Just gonna make sure things are actually correct here. I'm sure there is a smarter way to set up prior to stream so that I don't have to do it while I'm on camera, but I don't feel like if I do that, I'll just mess up anyways. Almost nothing is automatic on your channel. That would take time. I like investing the time into figuring out clever or not so clever ways of doing things so that they become automatic. I don't know. I just have way too much fun with my stream deck. My Sammy stream deck. This thing. I feel like I saw... Yeah, you have a streaming and tech section on yours, I believe. Maybe sometime I'll post my code in case anyone's curious about it. Although most of it I actually originally, not most of it, I originally got the idea from Dietom's Attack, that's it. You spend the time over summer or breaks to play around. I see. That's a good time to do it. I have too short of a memory though to do that, so I always have to make sure that I really do things when I have the motivation and memory to actually do them. Otherwise, I'll never get to it. Okay. I need a new tab. Oh, you saw us chatting about on the Knowledge Foundation? Yeah, that was a while ago. I basically was wondering if Jeff Bezos's really, really cheap Devil's Deal Amazon Fire tablets would work as a streaming tab or a stream deck. Sure enough, they do work, with the caveat that you should not buy it with your account, because then you will have ads and you can't... Well, I don't know if I should say this on Twitch. I'm sure you can. There's ways around it. I purchased legally under my account my Fire tablet. But yeah, you can really easily use it as a streaming deck. It's nice. It controls my lights and various things like this. Boo! So worth it. That was definitely worth the like half an hour it took me to program that in one day. But yeah. All right. So, which side is it? Riker. Vanguard, you are not late. You're absolutely not late. If anything, I am late because I was trying to figure out why speaking of Stream Deck, my Stream Deck was not showing me how many current people are watching. Didn't solve that, so I just decided to start. Also, you're late for being early. You're normally very, very early. All right. You were playing No Man's Sky and lost track of time. Fair, fair. Did they just came out with an update, right? Or they've announced a new update, like No Man's Sky version 4 or something like that. I have to say, I don't. It's not quite my game. A little grindy for me, but it's a wonderful game. I could never fault them for what they've managed to put together. Oh, it came out under a week ago. Awesome. Actually, No Man's Sky could be fun to play. I really would love to learn about their like creature generation. On the sort of general long-winded arc of my stream, the idea of artificial life, alien life, predicting life, all this stuff is kind of wrapped up in how we like spore, which is another thing. No Man's Sky, these procedurally generated or just user-generated organisms within a framework. They added a fully customizable difficulty setting with the new update. You'd love that. You'd love options. Nice. Nice, nice, nice. Yeah, I saw that too. That's cool. Well, okay. So, normal stream tonight, at least planned. However, I did get my flu and COVID vaccine today, and I had flu-like symptoms after my 3 previous ones, and I can kind of already feel it. So I've got my light hoodie on and a t-shirt underneath to keep me warm in case I start getting cold and shivery. I'm going to self-medicate with alcohol, which I've been assured by a physician who is board certified that that's not a bad idea. At least not a dangerous idea. A normal stream, there's no such thing. Banned. No, you're right. I don't think anyone has a normal stream anywhere. Ancom Psycom, hello. Yeah, you love this episode? Creepy as fuck, but the de-evolution idea was interesting. Yeah, so actually, I was supposed to have a guest on months ago who ghosted me to come talk about de-evolution and evolution in general. I'm going to have a different guest on eventually. We are going to talk about this. But I just thought this was going to be a funny image to start on. Because I did want to talk about, I want to bring in like the sort of, not evolution, but this one was an interesting case of the idea of ancient humans in science fiction. But I wanted to talk about Svante Pääbo and his winning the Nobel Prize today, and ancient Neanderthal and ancient human genomes, and how we're all, not all of us, but many people can trace ancient ancestry to extinct humans. You need an international standard stream for comparison. What would that be? What is the standard stream? Does that mean it has to just be the most cut and dry stream? I'm not really sure what that would be. Or would it be like a negative control, like the when we go and watch like MST3K or like the channel that streams just turtles, like the content is great, it's consistent, but it's kind of it's like a baseline. In that case, I'm far below it. I don't know. Okay. Yeah, I also love this episode, by the way. It's crazy. It is ridiculous. I love the concept. They also use big science words that they definitely didn't look up. But it was what was this one? I have it right here. Season 7. Okay. This was well into the 90s. They should have known better. They could have looked this up. But that's okay. All right. So, before I get sidetracked, I at least want to introduce the person, not who directly I want to talk about, but who did a lot of the work that I want to like mention, which is Svante Pääbo. Svante Pääbo, who is the Swedish scientist out of Germany, working at the Max Planck Institute for Evolutionary Anthropology. And he has been doing amazing work in collecting, sequencing, and analyzing ancient human genomes basically he's the guy who does it. So they've done some really cool work over the years. Spot turning into an iguana was hilariously bad. The whole thing was hilariously bad. I mean, I would say Barkley turning into a spider was even more bad, but I agree. At least Spot kept the same number of limbs. So happy that you now know you're probably part Neanderthal. I mean, I am going to make assumptions, but you have European ancestry, yes? If so, not just Northern European ancestry. If you have European, Asian, Pacific Island, and indigenous Native American, those populations, which is the majority of humans on the planet, then yes, you probably have some Neanderthal ancestry. The only populations that don't have don't historically, it's different with humans moving around over long periods of time, especially after the advent of modern travel. People with African ancestry don't have much Neanderthal DNA, and even that, it's like Saharan and sub-Saharan. Northern African populations do have Neanderthal, and then Denisovians are primarily Asia and then further east, so Asia, Pacific Island, and then indigenous Native American. Let's see. Oh, Miss Rizzo, hello. I missed you pop in there. You love Barclay's and scattered varied character arc. He wasn't on a ton, but he got to do some really cool stuff. Yeah, absolutely. I liked his character arc, continued character arc in Voyager, actually. Yeah, so Asian populations and over have Denisovian. So Denisovians and Neanderthals, I believe, also interbred. So let me actually see if I can find it. This is a great, fantastic just transition into talking about some real science. I have a bunch of stuff up, but I don't think they actually show the family tree that I'm thinking of. All right, let me look it up over here. Denisovian. I think I've been pronouncing that wrong. Neanderthal family tree. Where are ya? Svante Pääbo, by the way, gives a fantastic talk. I really recommend checking out basically any talk he's given that is on YouTube. I had the pleasure of meeting him in person after he gave a talk when I was in grad school. Super cool guy. Very, very cool. All right, I have no source for this, but this looks good. Yeah, modern humans. So they have it kind of, they ordered this kind of weird, but it's geographic, right? So who left Africa first and became Denisovans and Neanderthals? Was that Homo erectus? Oh, I actually don't know this. Neanderthal, Homo erectus, or are they? Let's see. I'm just trying to figure this out. I guess it's not that important. Regardless, Denisovans. So Neanderthals were all the way from France all the way to what is now the Middle East. I guess what is now France to what is now the Middle East. But that border seemed to change. It moved, I think, into modern China, and it even came back to the Caucasus Mountains as well. Like they moved around quite a bit, or their range did. And Denisovans were kind of like on the other side of that, so they were more on the Asian continent proper. So like sub-Siberia, China, and they probably, I don't know. I don't know if they moved to any of the islands in the Pacific. So they were there hanging out. There was a border between them. And so this graphic is denoting that, some of the Neanderthals interbred with Denisovans. Meanwhile, modern humans came out of Africa. So that's why this red arrow right here is pointing to everyone above Africa. That's because, some humans stayed in Africa, others moved out. We moved out, met the Neanderthals, and so everyone directly out of Europe has Neanderthal ancestry, and then the people who then continued to move on towards Asia, Oceania, and the Americas eventually encountered the Denisovans as they moved east, and we interbred with them as well. Okay, I gotta catch up with chat already because I was just talking. You love the episode where the Cytherians turned him into a super genius. That was a fun adventure. Yeah, that also, like, I love that one. That had some of my favorite Star Trek tech in it too, when he started going kind of wild. Paralleling the Stargate Atlantis episode, where McKay was turned into a super genius and invented a lot of math that didn't make any sense to him later on. Love that sort of stuff. You love talking about other hominids. I'm so glad you're here. They aren't even here to defend themselves when we talk about them. yeah, they never show up when we talk about them. What does the I stand for in the initialisms? That's a good question. Let me check where I found this. I assume that's like a place. So Vindija, and Altai for the Neanderthals. I know those are places. Same with Denisova. They are named after the place in which they were discovered. I am trying to figure out, though. Reported the results in Nature, and that's a broken link. Awesome. I bet if I just look for Denisovan map. Denisovan map. Map. Yeah, the Denisova is the Denisova cave. I don't think we actually have... I don't know how many representative samples we have of them. Yeah, Ancom, I'm sorry, but I'm not finding what that I is supposed to mean. I'm going to assume that it is named after... Oh, same thing with the NI. That they're named after some region or the sample that was taken. I kind of assume D stands for Denisova and N stands for Neanderthal, but I can't figure out what the I means. Not island, that's for sure. D.I? Okay. Denisova cave. And you know what? Actually, now that I'm thinking about it more, I believe in multiples of these places. Well, I'm trying to look it up now too, because now you've got me curious. So in a couple of these places, and I think the Denisova cave in particular, there are different substrates. So I wonder if that's like different levels of the cave, right? Because there's, you know, a time-based buildup of material. So the further down you go, the more ancient you get, or something like that. I wonder if that's it. Let's see. D.I. Denisovan. It's not something that Google likes to pick apart. Individual? Ah. That could be it. Also, hello, Rizanabe. Welcome. I was very excited to be talking about my sequencing results earlier today on Discord. I'm happier with the results, but it's very suspicious the way, you know. Yeah, I think so. Denisova individual, Neanderthal individual. That's what I'm seeing. There you go. All right. So, speaking of my genome, though, for any... Oh, I need to mention a couple things. New community goal that I started. I was encouraged due to my successful question mark sausage making venture to do more cooking. I refuse, at least for a little while, but I do have this Japanese candy kit where you make all these little gummy pieces, and it makes pineapple, strawberry, banana, orange, chocolate flavored cheeseburger with bun and ketchup, French fries, and cola. So if we hit that goal, I will do a intermediate baby cooking stream. You've done one of these kits that can be kind of weird if you're not used to Japanese candy flavors. Yeah, well, I was even confused that I wasn't even sure what they were supposed to be flavored. I was confused why ketchup had a strawberry next to it. And I am not expecting these to taste very good, but the process looks fun. I'm not gonna be able to share this very nicely. Yeah, I guess it looks fine. There's a lot of steps. It looks like a lot of fun. So we'll do that when the goal is hit. The IGV window looked a lot more like 30 coverage. Yeah, I haven't calculated it yet, and I will get you that header. I did see the header, by the way, of one of them. I'm curious what the instrument they used was. I do not know them off the top of my head, and it started at A00, and I couldn't quite figure out what that is. So if you happen to know what instrument that was, or just wait till I give you the header to that. James Gurney, welcome. Yeah, everyone should go follow James because he recently streamed from his brand new lab, and that was fun to watch you set up. Also, very jealous of your view. It looks very nice for a scientific lab. Let's see. Okay. I'm going to laugh every time we go back to that. All right. So I don't even know where to start. So Svante Pääbo, he won the Nobel Prize this year in whatever biologists are given. Is that physiology? I don't even know. And he got it for his work collecting, decoding, and analyzing ancient hominid DNA. Oh, Rai Saruni, welcome. You also like this episode? Everyone's liking this episode. It is a lot of fun. So his work is kind of I wanted to like think about / talk about. It's just super cool, and there's like a historical basis to it too. So we can go from, and I should have put these in chronological order probably. I think this is it. Yeah, even in what is this, 1989, he was doing work to extract ancient DNA via PCR, which back then you would spell out and put in your title because it was cool. And I honestly don't know where this idea came from. I'm just trying to find a representative figure. This one isn't even all that cool because they started on mummies. He started by trying to extract DNA from mummies, and I don't think it's this paper, but eventually, it's funny, his first results, which I think were published in Nature, which was the ancient DNA of a mummy that they found. Not them, but they had on hand. Later on, it turns out that DNA was actually not from the mummy. It was actually probably from some museum curator at some point. Because they realized this very simple fact, which was that DNA molecules, they're linear, they're long, they're, depending on what organism, tens of millions, hundreds of millions of bases in length for a single chromosome. But you expect that over time they would break apart. That just happens. I forget what the half-life of a DNA molecule is. But it's not infinite. And so what that means is just over time, these molecules break into smaller and smaller pieces. And that provides a pretty difficult time genomically. And so what do I mean genomically? Here, let me actually pop this over. So this is me. This is my new data. I mean, that is a lot better, right? So if I zoom in here, so right here we're looking at, you know, 241 base pair, so 241 letters of my DNA, but it's still so zoomed out that we can't even see the individual bases. Oh, there they are. Maybe we could see them and I just didn't notice. Yeah, they're down here. And these pink and blue things here are what we call reads. So these are individual strands of DNA or fragments of DNA that have been sequenced. What sequencing means is you just read the DNA, something we can do now fairly trivially, but back in Pääbo's day, absolutely not trivially, especially when we didn't know where these would go. So these are sorted by where they exist on the genome or where you expect to find them. But basically they realized that, oh, they got this nice mummy DNA, but then they looked around and were like, wait, this is too big, this is too long. Because in reality, they expect little itty bitty tiny fragments of DNA to pop out of ancient samples. So I think, yeah, that's this one. Okay, so in 1997, they did the really cool thing of grabbing Neanderthal bones, so not fossil, but bone, because they had not completely been mineralized, and they began looking for DNA. And I love their figure 1 because it probably would make an anthropologist's heart skip a beat, because they came in here with some sort of instrument and sawed through the right humerus of a Neanderthal specimen. I don't know how many these would be. You know, how many like samples they have of Neanderthals, but it's not a ton. It's not like we have, I don't think we have even in the dozens of complete, or not even complete. I don't know how many individuals versus complete skeletons we have. We probably don't have any complete skeletons, but we have near complete skeletons. Suffice to say, there's not many humeruses going around, probably, especially not in 1997. David T. Gregory, welcome. Hello. Cheers to you. Actually, in celebration of that, I will cheers with my beer, which is now overflowing. It's very, very good. If anyone's ever in North Carolina, get Rocket Science. It's very good. All right. I am drinking Rocket Science by Full Steam Brewery from North Carolina. It's an IPA. It's very, very good. I don't like IPAs normally, but I really like this stuff. Just water for you. I also have my water over here because my throat gets dry. Am I on Untapped? I have been on and off Untapped probably since I was like 20. So over a decade. I believe I am currently not on Untapped. The problem is I have too short of an attention span to reliably take a picture, find, and then rate things that I drink. And I try too many things too often to ever kind of double back on something, unless I find it at like a grocery store. But if you have recommendations, recommend them to me. I'll drink them. You can't stop me. All right. Also, I'm just checking the bitrate here, which has been my enemy recently. It's looking good. Someday my bitrate will be amazing when Google Fiber stops canceling every appointment I make with them and gives me Google Fiber. Okay. Anyways, okay, so they did some. Let's figure out what they actually did, because they did some not crazy things, but they took this bone and did very scientific things, but it also still sounds kind of scary. They removed remnants of varnish, which were used to preserve the bone. I assume that's a little bit of the shininess here. Then they hydrolyzed the samples under acidic conditions to release amino acids that were then analyzed using some biochemical techniques. They're just looking at some proteins. We'll kind of ignore that because actually I care about the DNA more. So they extracted DNA from 0.4 grams of the cortical compact bone. What is the cortical compact bone? Cortical compact. So that is the outer layers, it looks like. It's been a while since I took anatomy and physiology. Don't remember my bone parts. Yeah. Oh, that's interesting. For some reason, I was going to assume that it was the soft, squishy parts on the inside. Okay, shouldn't that treatment pretty much ruin everything? Okay. It's not like they dipped it into like a vat of acid. Proteins are chemicals. DNA is chemicals. So I'm sorry, but you're filled with chemicals. And by being smart about what chemicals you... varnish plus acid doesn't sound very good. Well, they removed a lot of the varnish, apparently. So if you're really careful, you can extract different biomolecules using different conditions. So, for example, in the lab, we often use a mix of phenol and chloroform in order to separate out proteins from RNA and DNA. And then to further do something like wash that DNA or get DNA, you would add in isopropanol. So you're adding things with different chemistries, that will not necessarily harm the proteins or anything like that. Not in a way that would destroy them. Make them non-functional, probably, but not destroy them. So we have, oh boy, at this point, many decades of knowledge of how to extract these things. All right, so they extracted, but what I'm impressed with is using the best of 1997 technology. They were extracting DNA from 0.4 grams of this bone. So previous experience shows that ancient DNA tends to be degraded and damaged to an extent that makes amplification of segments of mitochondrial DNA longer than 1 to 200 base pairs difficult. So this comes down to the simple fact that, like I said, DNA degrades over time. They're just assuming that ancient DNA is pretty degraded, like pretty much the same, like give or take probably a couple thousand years. It's probably about the same. Then again, maybe it's not. So amplification relies on these things called primers, which are basically, hey, start here and also start on this side and make everything in the middle, everything being all the DNA in the middle. That's a lot easier if you have a big long piece and you can kind of guess where those ends are going to be. If they're little tiny, it is kind of hard. But they apparently did find some that were able to amplify a 105 base pair segment. So they did that and just got DNA, so that's great. You see, yeah, you thought no longer functional equals basically destroyed. Yeah, I would say not a rule of thumb, but a good way to think about it is that proteins can be made non-functional and destroyed. So for proteins, structure basically is the function. So if they're not shaped the right way, it's still a protein and you could figure out what it was, but even though it's not going to work. It's like a car that's like totaled. you can probably still figure out what it was, even though it doesn't roll, right? But it's not technically completely like obliterated into its component parts. Maybe that's not the best example because you could still probably figure it out. Okay, so they did some PCR. I think that's, oh no, that's not it. They don't have it. But they did this very, what at the time was probably very difficult. So they have quite very little to go on. So up here at the top, I need to zoom in even more. So what they're doing is they took these pieces of DNA that they found, and they were basically doing it like we do now, except with much lower throughput, being able only to sequence I believe 4 amplifications, and I'm not really sure what these are, if they're replicates, dots, blah blah blah. Yeah, so they have this reference sequence up here, which is the human genome. And the very cool thing that they found immediately is, oh, and I don't know what these stars are, is sure enough, but the mitochondrial genome from these ones, so like A10, A17, B11, have these sets of differences. So the letters are what are different, and there's some odd ones here and there, but we can pretty much we can believe the ones that are consistent across them. Sure enough, the human and Neanderthal genomes, mitochondrial genomes, are very similar, but they contain differences. This is like such a small figure for such a huge finding at the time. It matches expectations, basically. We knew they were human-ish. We knew they were related to humans, but you don't actually know molecularly until you go look. Kind of like sifting through garbage to see what someone had for lunch. A banana peel and some chicken bones are pretty telling, even if you can't eat them anymore. Sure, yeah, that's a good analogy. That's actually a lot of what like biochemistry often is. It's nice to get the whole chicken out, but sometimes you just have to settle for a leg bone and you'll figure something out. In fact, that's probably not just molecular biology. That's probably most science, come to think of it. All right. Oh, yeah, and then so in this paper, okay, I'm not going to be able to flip this. These little chunks right here, as we go across, this is really cool. It's silly, but it's very cool. So we have the reference sequence up here. So now we're looking at a much longer sequence of mitochondrial DNA. And, well, you can't make those big of sequences from this little tiny Neanderthal DNA. So they're doing something not dissimilar what we still do today, which is they are sequencing it in chunks. So they have these little chunks right here and these longer ones, and then some that overlap. And what they're building out of that is what's called a consensus sequence. So that's this down here, which is really hard to see on screen, I realize. But it's basically saying, hey, look, Neanderthals, they have a G here. They also have a G here, a C here, a TT here, a TT here. And they're building pieces of the Neanderthal genome. That's more or less what is going on here, except that we know that me, should be Homo sapien. I should be a Homo sapien, but there's still differences. So like right here, A is the worst one actually because of that color. Let me find another one. So I, for example, at this position, even though the reference sequence, the sequence that we expect, has a G, I have a bunch of T's. Because apparently one of my parents has a T there, and the other parent has the G. And so that's normal. Uh-oh. That's very normal. I am a very normal human. So if I zoom in here, these are the things that are called SNPs, single nucleotide polymorphisms. People differ in their SNPs. It's kind of what makes us, no, it's not kind of, it's exactly what makes us unique. It's just that when you were to examine the difference between a Neanderthal and a human, you would see many more differences. If you go farther away and go to something like chimpanzees, not only would you see more differences, you'd see structural differences. Genes or even whole chromosomes might be in different orientations, is the way I'll put that. Wait, why do I have a new comment on Instagram? Something I don't use. No, it's nothing. Okay. Yeah, so Svante Pääbo. We don't care about that. And then eventually, in 2016, they sequenced the whole Neanderthal genome, which is obviously took a lot of work. I really, really am curious about how they determined who were going to be the first authors. There are 5 first authors. I wonder how. So they're all, wait, let's see. Max Planck. So they're all the same university. Oh, wait. Actually, is the one? Oh, no, that's what the one is. Members contributed equally. So that is the hashtag. Oh, okay, so there is one. So Richard E. Green got the credit for first authorship. Okay. So sequencing of Neanderthals. Oh, they spell it without the H here. That's interesting. So Neanderthals are our closest evolutionary relatives, or at least the most famous one. I don't remember. So where's that picture? Denisovans, I think, are split from Neanderthals, but just as related to us. For some reason, I was thinking there was another one. Maybe not. Maybe it was just Denisovans. But, science is cool just for science sake. It's really cool to, think about how we are similar to these organisms that lived and died thousands of years ago. And the fact that what we learned after sequencing, actually, we kind of knew this before, but after we sequenced the full genome, we became very sure. I don't think there are any figures here. No. We're very sure that we interbred with the Neanderthals. Troglodytes? Not quite. I think that's I don't remember what the word troglodyte means, but that's like chimpanzees are like pan-troglodytes or something like that. I'm trying to figure out. Let's see. Sweep screen. I think they found out. Whoops, I keep clicking on things. Oh yeah, they even identified SNPs, which is cool. That means they had enough allele variation. Let's see, I'm not sure what selective sweep is. I'm wondering if that is... see, what I really wanted to find was a list of Neanderthal SNPs, which does sort of exist. It's just that some of them are in kind of boring, like they aren't actually in genes. So wait, amino acid changes that are fixed in present-day humans but ancestral in Neanderthals. Gene starting, blah, 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 blah. Yeah. That is actually what I kind of have up on this other screen, though, is recently this came to a, not a head, but just like a interesting thing, which like an oh, by the way. A recent genetic association study identified a gene cluster on chromosome 3, which was a risk locus for respiratory failure after infection, with severe acute respiratory syndrome coronavirus 2, SARS-CoV-2. So they found that this risk allele is conferred by a genetic segment around 50 kb in size that's inherited from Neanderthals. And you can't see all of this. And it's carried around by around 50% of the people in South Asia and 16% in Europe. So this region that I'm zoomed in on my own genome is that region. So it is the risk allele region. So I actually haven't looked at this yet. I want to know whether or not they list these specifically. Yeah, so what gene is it? It's I just had it. LZT-FL1. LZT-FL1. Leucine zipper transcription factor like 1. So what do? A gene encodes a ubiquitously expressed protein that localizes to the cytoplasm. It interacts with some other proteins, regulates protein trafficking to the ciliary membrane. Okay. But what are... Whoop. I forgot he was there. He scared me. So I want to look that up, but I also remember the Miss Frizzle, just because I know you're interested in sort of, even though it's like the spermatogenesis, the reproductive side of things every once in a while, there is a set of alleles or an allele that some people get from Neanderthals, like it's from the Neanderthal genome, but humans who do not have Neanderthal ancestry don't have this. It is a gene that changes a receptor for progesterone. And let me see if I get this right. If you have this Neanderthal allele, let's say, your response, and also you are able to have and carry children, like you have ovaries and a uterus, what happens is that you are less likely to have a miscarriage, but there's a genetic trade-off. You are then more likely to have a premature birth, basically. So there's a little bit of an evolutionary trade-off there, and that is derived from a Neanderthal gene. Now, I don't quite remember what the genes were, but I remember when I saw Svante Pääbo talk, he had mentioned how it's very clear that certain regions are under selective pressure in modern humans. Certain regions with Neanderthal DNA are under selective pressure. So when he or I say that, as a European, Asian, North American, Pacific, or just American Pacific ancestry person, you have 1 to 2% Neanderthal DNA. That 1 to 2% are in specific places, because that's what evolution does. We interbred. Evolution was like, hey, those are useful, and then fix them in the population. So, like, for example, I'm not sure this risk allele, though, is clearly not useful, but evolution doesn't know about future diseases, so it's not its fault. Let me see. Neanderthal chromosome. I want to say I saw a picture of it. No. There's like segments on like chromosome 9 or something like that. I'm not gonna be able to find it. This is just from literally the same thing we were just talking about. Ooh, yikes. Yikes. What is this? Modern men do not carry the Neanderthal gene? Oh, okay. Ooh, I probably shouldn't have accepted those cookies. Is anyone here? Well, James, you're from the UK. Although, is the Irish Mirror a reputable news source, if you're still here? Do you know anything about this publication? Modern men do not carry the Neanderthal Y chromosome, a new study has claimed. Our ancestors and their Neanderthals neighbors may have been kept apart because they had incompatible male genes? What? Okay, hold on. Despite the evidence of interbreeding, I'm going to have to look at this paper now. Despite this evidence of interbreeding between the two different human species, with today's humans possessing 2.5 to 4%, researchers from Stanford University, okay, if I click that, it will just tell me about Stanford, have claimed it is telling that no one contains the DNA from Neanderthal male Y chromosome, which is only transmitted from father to son. Why is that telling? Huh. That PGR piece of trivia is interesting. Part of what causes its effect is an alu element that is retrotransposed into the area. And you're not sure about the Irish Mirror. I mean, I figured. I don't mean to tell you to speak for, you know, a whole media atmosphere. Article looks sus. Yeah, that's why I'm reading it, actually. Yeah, so alu element of the retrotransposed on into the area. Yeah, I don't know the exact details of that. If you have a link, that'd be cool. I'd love to read more about it. Blah, blah, blah. One reason that the Neanderthal Y chromosome genes are incompatible with other male... Oh, could be that Neanderthal Y chromosome genes are incompatible with other human genes. They may have led to miscarriages, prevented the birth of hybrid male babies. Okay. As a result, Neanderthal Y chromosome would eventually have exited the human gene pool. I mean, that also can happen due to just genetic drift. And also, the Y chromosome doesn't recombine, so you would either lose it, or we'd either have it or we wouldn't. We've never observed the Neanderthal Y chromosome DNA in any human sample ever tested. That doesn't prove it's totally extinct, but likely is. That is true. Okay, I need this paper. The research is reported in the... Okay. Oh, thank you. They didn't actually cite the paper, so I gotta search this Neanderthal Y chromosome. This is 2016. Could be this one. I bet they're just kind of misreporting it. Or not misreporting, but misrepresenting. All right. So this is much better. Suggests that the Neanderthal Y chromosome genes disappeared from the human genome long ago. Yes, okay. Look at him. He's beautiful. Max Planck? A researcher at the Max Planck Institute for Evolutionary Anthropology also co-authored the study. I wonder who that was. Okay, this is sounding less sus. That article is just maybe spinning it in a weird way. Okay, so, ooh, a 120 kb of exome captured Y chromosome DNA from Neanderthal individual. Nice. Wait, was this after the draft or before the draft? Genome. I misread this. I thought this came out in 2016. Oh, this became available in 2016. It was published 2010. Get out of here. All right. We identified protein coding differences between Neanderthal and modern human Y chromosomes, including potentially damaging changes to these proteins. Three of these changes are missense mutations in the genes that produce male-specific minor histocompatibility antigens. Antigens derived from that, for example, are thought to elicit a maternal immune response during gestation. Wow, that's actually... Okay. I'm a little bit more of a believer. At least with these sentences. I guess if you just say it smart enough, I believe it. Which is not good. Ooh. Math. No thank you. Oh, we can look at these. All right, these are human. No, these are Neanderthal. I could take my data and align it to the Neanderthal genome. That'd be fun. That would just throw out a bunch of reads, but you know. Let me grab this progesterone paper. Look, it's our friend, Svante Pääbo. Whoa, okay. All right, oh, super archaic. Interesting. Okay, so effect. Lurking for a bit. All right, so we have a couple genes that we can look at. I can actually search these genes. That's a little tough on IGV because it doesn't let me search as easily as I would like. So we get just some substitutions. What if I have the Y chromosome from Neanderthals? Where's my Y? Where's my Y? Oh, liftover. You're smart. Yeah. Yeah, that's a good thinking. See, my current lab is very anti-USC or UCSC genome browser, only because of Drosophila things. Don't worry about it. So I've forgotten how to use it. But good point. UCSC genome browser. So smart. Human. Would it be under human? Okay, well, I'll go to human. Yeah, I'll go to human. I'll go to human and see what leftover they have. Let's see. And I'm going to need HD19 because it's still HD19. Just lazy. Ain't no. Yeah. Most of my bioinformatics and genomics knowledge comes from me being like, I want to make this thing happen quicker. So it is it under tools? Yes, it is. Human to Chinese hamster? Crab eating macaque? No. I wonder if they have it. Oh, they have the T to T. Yet another thing I need to do. Maybe they have it leftover from another one. They don't. Sad. Okay, well, it was a good thought. Oh yeah, it is under tools. Yeah. Well, next time. Maybe if I just search human to Neanderthal genome coordinate. It's rough to use, but lots of interesting stuff. Yeah, I'll have to hit you up on Discord. We don't have a T to T completed project yet for Drosophila, so I haven't had to deal with it yet. So if you have any tips on how to set up my my mapping program so I don't completely just mess it up. I'd love to hear it. All right, I'm just gonna have to whatever. I'll just have to map it to Neanderthal. Map myself to Neanderthal. Did I missed chromosome Y? There we go. All right. Our little tiny chromosome Y. The sad little chromosome that could. So this is ZF. Better do expanded, please. I believe the article mentions ZFY. And that's the wrong side. So since the Y chromosome doesn't recombine, I'm going to assume that I just have to look for a single SNP or a single change, and I'll know whether or not I have the only example of a Neanderthal Y chromosome. The difficulty is in the annotation mainly. They use their own gene annotation, so you end up looking at gene names. Ugh. No. We have enough annotations. I truly hate that. Why do they do that? Also, is that like chromosome mitochondria 1, or is that just some random number? And that's awful. I hate it. Thanks, I hate it. It's truly frustrating. I mean, someone has got to have lifted over the coordinates in like some annotation file, right? Right? Please. Okay. ZFY valine 140 to alanine. I can roughly estimate that. So, you think it's just a random gene? Well, still. How big are we talking here? So 600 divided by 3. So that would put me right about here for 140, valine 140. A lot of valines. No SNPs. My Y chromosome is unfortunately Homo sapien. That's okay. That's okay. All right. Speaking of which, though, should mention the Sci-Fi. So, multiple people apparently's favorite episode, or one of our favorite episodes. So this episode is interesting. So it's called Genesis. So in this episode, a tiny whoopsie in some genetic manipulation causes it's so absurd that I have to like. Oh, something just started on my music. Okay, skip that song. Oh, it won an Emmy. Interesting. Wait, they saw 10 Emmy nominations for outstanding individual achievement in makeup for a series. Okay. Sorry. So it's yes. So Barkley has a mild case of the flu, and Dr. Crusher injects him with a synthetic T cell to activate one of his dormant genes that confers immunity. Okay. No, but, okay. This T cell apparently becomes like airborne or something like that and starts infecting other people and turning on their dormant genes. But it's not just dormant genes. What is the website with Star Trek scripts? Star Trek script. Here it is. Chakotea. Chakotea. Okay, I need to search. Oh, whoops. Actually, not the cage. Genesis. I think they said something about introns, which is interesting. Chakote. But it has an A at the end. It has an A at the end. Also, hello, Maddie. Welcome. Introns, yes. I have analyzed Commander Riker's DNA structure. A synthetic T cell has invaded his genetic codes. This T cell has begun to activate his latent introns. Cheers. Also, whoa. Hello, Embis. Haven't said a word, but have already redeemed some points. I love it, by the way. Introns, says Picard. They are genetic codes which are normally dormant. They are evolutionary holdovers, sequences of DNA that provided key behavioral and physical characteristics millions of years ago, but are no longer necessary. For instance, Counselor Troy's gill slits and other amphibious characteristics were derived from introns, which still contain amphibious codes. Picard. So these introns are causing her DNA to recombine into an earlier configuration? That is correct. In her case, the DNA has created an amphibious life form, which became extinct over 50 million years ago. And Commander Riker's introns are changing him to turn into what appears to be one of the earlier hominids. Yes, sir, I would say Australopithecine. Oh, that's something we can search. Australopithecine. Because that's not what Australopithecines looked like. Here we go. Figure A, figure B. A, B. So they just say Australopithecine to, grab the larger, what is that? The sub-tribe I'm seeing. So, yeah. So I don't know, in like 1995 or whenever the episode was, I don't know what had been discovered by them. It's a genus? Okay. Oh yeah, Australopithecus afarensis. Yeah, okay, there we go. Afarensis? Yeah, afarensis. They'd found Lucy by then. Okay, well that's I bet that's what they were referencing, but they just say Australopithecine. Like I'm just looking at the script, which by the way, each of these stages is another link in the evolutionary chain, which stretches back to the origins of all life forms on Earth. Which is when as we all know, an ancient group of hominids, or not hominids, ancient groups of humanoids who were completely bald came and sprinkled DNA over everything. Because the introns can include genetic material from many different species over millions of years of evolution, it is possible that a wide variety of transformations is occurring among the crew. I.e., that is the sentence that they added in to explain why Barclay is a spider now. Oh, actually, they keep going. I love stuff like this. What about those crew members who are not from Earth? All humanoid life has a similar genetic pattern. The virus should work on non-human crew members in the same way. They are each de-evolving to earlier forms of life on their homeworlds. Okay. That was actually a huge lampshade. You're huge into biological sci-fi. Absolutely. I love biology sci-fi. And yeah, you're right. That's not how evolution works. But I love it when they use the words. It's like a toddler learning how to speak. I like it when they use the right words, even though they're not in the right order, or they don't think they mean what they think they mean. It's a lot of fun because introns are not ancient what they say, evolutionary holdovers, sequences of DNA that provided key behavioral and physical characteristics millions of years ago. I mean, like maybe an intron contains an intron, for anyone who doesn't know, proteins are the basis of life. They provide structural and biochemical functions, right? Okay, but we got to keep those proteins encoded somewhere. That's encoded in the DNA. The DNA has a lot of grammar, like Maddie is saying, commas and semicolons. That DNA grammar contains bits of DNA that actually aren't really used, but are still kind of there. So you pop from one part of the gene to the next part of the gene, kind of skipping the part in the middle. It's actually cut out of the RNA that is made from that DNA, and those are called introns, the thing that's cut out and thrown away. Now, they actually do have a function. That bit of RNA that is chucked out can actually be used for regulatory information. And the existence of introns is essential because the existence of splicing is required for the diversity of proteins that we make from our DNA code. You can basically imagine if you start, they're hot swappable. You can swap them out in a limited fashion, and evolution can be driven by the new addition, retention, or loss of bits of DNA, not because the DNA is missing, but instead because they're included in an intron or provided from an intron, what was formerly an intron, and is now an exon. But that does not mean if you suddenly use my introns, you will get an Australopithecine. Instead, I will probably die. It's a bad thing. So if we grab any... Here, wait. Back to my genome. Let's go to... I bet if we just... Actually, we're going to go to the genome browser. UCSC, because I know how to use at least this part of the genome browser. Genome browser, human, go. Let's just go to some ACE2. Hey, that's important. We're just going to go to full base position. I hate how many introns the human genome has. I'm going to go ahead and zoom in right there. Flip that DNA right around. Yeah, so right here, this gene, ACE2, is very important. It also should sound familiar because it's important for infection by SARS-CoV-2 to cause COVID. When we make a protein, we start from this little green block right here, follow all these little gray blocks, gray block, and then we actually skip across off screen. However, because this little line part here is an intron, so normally that's removed. But if we were to retain some intron and keep going, we would hit this little red thing, which stops the protein being made. So in fact, if I were to retain my intron, in this case, rather than having a nice big ACE2 protein in order to do whatever functions it does in my mucous membranes, because it goes from this bar to this bar to this bar to this bar to this bar, etc., it would stop early and probably die. You'll probably just get a bunch of random retroviruses that don't work if you reactivated your introns. That sounds very boring. Yeah, that too. So introns themselves are hypothesized to be ancient retroviruses. I believe because the spliceosome has similarities to a class of retrovirus or retrotransposon or something like that, which is an interesting thought that eukaryotic ancestors were infected by genomic invaders a long time ago. And as a mechanism to get around it, we just kind of ignored those sequences. But then we used the components of those genetic invaders to our advantage and evolved the intron-exon system. I know that's a hypothesis. I don't know how much data there is for that. Whoever presented that to me once was very convincing, though. But yeah, also we stick a bunch of other random sequences in there. All right, Rizanabi, I saw that you linked me something. Here's a link to some genome browser tracks that have Neanderthal reads mapped to the HG19. There's a track for Neanderthal SNPs. Thank you. Can't find NT-seq read in track database HG19 chromosome X. Is this just a track that's on there? Like our cells having originally packed this. Sorry. I'm going to say that. Next time I have to teach about mitochondria, I'm going to use the word Pac-Man. Like our cells having originally Pac-Man mitochondria makes sense. Well, yeah, I mean, it wouldn't be the first time that our ancestral cells have utilized the biological functions of another organism. I mean, plants did it twice. So, track info page. I can figure it out if it's on here. But yeah, Pac-Man. Let's see. What if I just Control F, search for Neanderthal? Here we go. SNPs. Neanderthal seq. Let's check this out. So these are sequence reads. Yeah, which one were you trying to get me to do? So NT read seq. NT seeker. Was that this one? Here, I'll just do pack. Where'd you go? Actually, where'd you go? Which one was that? All right. We're gonna hide all tracks. So if you're not quite following what I'm doing, I am viewing the human genome, but I'm trying to see some Neanderthal stuff because I have my own genome over here that I would love to see. Back, refresh. So these are just sequencing reads? Yeah. What do I want to know about Neanderthals? Oh yeah, you're an anthropologist, right? Am I getting that right? Finally done with lab. Fantastic. Comparing my newly 30x'd DNA to Neanderthals? Yes, a little bit. Rizanabi is helping me grab out some Neanderthal SNPs. Ooh, Maddie. Oh, I didn't even think about this. We were talking about them genomically. Is that something you know about? Archaeology and forensic anthropology. Very cool. But you took a lot of paleo anthropology classes. The what word? Most of which is out of date. I mean, do you know, let's see, what was the full range of Neanderthals? That's what I was trying to remember. So they're in Europe. They're in part of the Middle East, but what was the furthest east that they ever made it, or that we think they made it? Because I thought I had seen that they made it almost into like what is now China, but I don't quite know. Oh, Brian has contributed 2,000 to the Poppin' Cookin' Challenge. Great. Thank you. I still want these snips. I'll put these S snips on here, these selective sweep snips. I'm not sure what selective sweep means. Oh my gosh, and alt misfrizzle. Ooh, I don't know what these mean. What is this? This track shows single nucleotide polymorphisms used in a genome-wide scan for signals of positive selection in the human lineage since divergence from the Neanderthal lineage. SNP labels represent the ancestral or derived status, determined by comparison with the chimpanzee reference genome of alleles in the human reference assembly. Okay. For example, a SNP labeled AAADAAA blah 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 has an ancestral allele in the reference human genome and in all modern human genomes except Han Chinese. Among Neanderthals, 2 instances of the ancestral allele were found, but no instances of the derived allele. Oh boy. Okay, that's confusing for me. I do not think about stuff like that. All right, Rizanabe, I'm curious to hear from you, but no pressure. We can figure this out. A lot of stuff has shifted to genetics. Yeah, we were talking to an extent about Svante Pääbo, who won the Nobel Prize for his work in genetics and genomics, characterizing, linking, and describing the Neanderthal genome and its contribution into our own DNA as modern humans. Yeah. Yeah, Brian, you met him too, right? He's a really nice person. His win was a fraction of what he... Oh, absolutely. I looked up his how like his papers actually here. Let's go ahead print. Oh, publications. Is this all of them? Yeah. I'm just scrolling. He has so much work. He seems like a total dork, LOL. From my recollection of him in person, he seems so sweet and genuine though. Very, very cool though. Ooh, an early modern human from Romania with recent Neanderthal ancestor. I remember this paper. His Neanderthal Man book. It's a great read. I will add it to a list that I am keeping. I don't know why this is all I know about his book writing, but is that the book where he comes out as being bisexual? Like one of his books, if he's written more than one, he comes out as bisexual. Let's see. Yeah, Brian, you had lunch with him. Oh, I'm jealous. I wasn't able to have lunch with him that day. I'm very sad. Rizanabe, you can't get the genome browser to load right now. Not sure what's going on. I think you'll find that the problem is that it is the UCSC genome browser, a useful tool, but nonetheless hosted on a server, not on your own computer or local network. That's why. Between him and Carolyn Bertozzi, yeah, some great human beings are winning the Nobel Prize. Yeah, absolutely. The other day on James Gurney's stream, he was mentioning Nobel Prize winner disease or whatever it is. You know, when some Nobel Prize winners tend to go a little off the rails afterwards. I wonder, Nobel disease, yeah. I want to know what Svante Pääbo's version of Nobel disease will be. I hope it's that he becomes a nicer person. An even more nice person. Ah, Maddie, you are sending me something. This is from 2019. Ah. There hasn't been too much of a big geographical shift on their distribution. Yeah, so I thought for some reason their distribution had like changed, moved from like right about here to right about here over time. But that, I don't remember where I even saw that. Also, I love that, yeah, also the Denisovans and Neanderthals were also getting it on. I like that it's not just us and Neanderthals. I'm remembering. I'm having like a memory, a core memory about Neanderthals. I thought that at some point someone had hypothesized that the reason why we mated so much / frequently with Neanderthals is that we found them very attractive. There was some scientific basis for that. What was that? If I just search Neanderthals were hot, will that come up with something useful? Thank you for the article, Maddie. I will. It sounds very Evo Psyche. Yes, it does. I have some very favorite Evo Psych persons. Evolutionary psychology. Encounters with archaic hominins. You think the selective sweep track is what I want. If you recall correctly, selective sweep is a SNP that is under positive selection. It is 100% penetrant in the evolved species. I will look more into that. I almost wonder if there's an easier track that I can just grab from somewhere else, that it just isn't preloaded into UCSC. Maddie, I'm going to drop this this link that you provided into the primary literature of the Discord so that I don't forget it. Australopithecines, cortical, browsing. Oh, thank you. Oh, did I see the video of him being thrown into a pond at his institute after he won? No. Here, I'll look it up and I'll show it. Svante Pääbo. Fountain? Wait, did you say fountain? Fountain, pond. Nothing's coming up. Thrown in water. Oh. Let's see. Just trying to find a good one. Okay. Whoa. That happened very quick. Here we go. And then I need to turn on my thing so you can hear it. Okay. Such a nice person. Okay. Ironically, you were taking an ancient DNA class when the Neanderthal nuclear paper was publicly released. It was like a bomb had gone off. What do you mean by nuclear paper? I don't know what you mean by that. I'll get... if I'm missing something... Oh, okay, nuclear DNA. Okay, I was going to say... Did Svante Pääbo discover that they had nuclear power? That would be the... Oh, That's his version of Nobel disease. He starts making claims that they were the dominant species. They're hiding. They created an alternative society underground. They left for the moon. Or my pet theory, which is that he and George Church get together and they breed a / genetically engineer a Neanderthal so they can talk to it. Before that, they thought there was zero interbreeding because their mitochondrial DNA didn't have any mixing. Admixing. What was I going to search for? I just lost my train of thought. So I'm just going to sit here for a second and think about it. Sit and think. It's gone. Whatever I was thinking was completely gone. But yeah, Neanderthals. Oh, right. I was looking at my own DNA. Duh. Yes, and Rizanabe, you were helping me out. But so I don't really understand this notation. I need to. It gave me the definition, but I need to like really think about it. I think what is better is if I just search for Neanderthal SNPs. Because I believe there is a group on Snippedia. All the paleoanthropologists were like, we have clear-cut evidence of fossils showing hybrid features. And then genetics people were like, you guys are trash and don't know what you're talking about. That sounds like genetics people. So the bones people won that round, but the genetics people had to learn a hard lesson that genetics isn't always perfect. Yeah, so, yeah. not surprised, not surprised. All right. Excuse me, there is a SNP right here that I see is a Neanderthal-derived SNP that says less likely to sneeze after eating dark chocolate. All right. It doesn't have a link, so I'm going to have to open that in a new tab. And I'm going to have to search this, I wish to know if I'm going to sneeze after eating chocolate. And whether or not I received that from my ancient Neanderthal ancestors. Chocolate makes me sneeze. Probably not. No, probably not as an allergy. Probably as a just a response. So like, I and many other people, if I go from a dark to a bright place very quickly, I tend to sneeze a couple times. I have heard it. I don't know the actual genetic, or not genetic, but the physiological reasons for that. You call them sneezers. Yeah, so you're that way, right? So the way it's been described to me, though, is basically like a, not a sensory overload, but a crossing of a sensory wire. So potentially, if it's like light receptors, sure, why not? Those are connected to nerves. Why not also smell or taste receptors? It's actually gotten worse for you where you're starting to bring it up with the doctor. Really? Oh, wow. Yeah, I know some people are just like generally more susceptible to certain light sensitivities than others. All right. Dark chocolate sneeze, which affects 25 to 30% of the population, is on chromosome 11. Okay. But what if you gave me the coordinates for that? I would like the coordinates. Oh. Ah, shoot. No. Oh, feeling Oh, this is a list I've been waiting for. Feeling hangry. Hitchhiker's thumb? Oh, that's when it's like super bent. Hoarding. Sweet. Ooh, okay. Blushing. Fear of heights. Yeah, here it is. Okay, awesome. I would really like to know, though, the coordinates of these. I can't just rely on Snippedia. Wonderful. This is wonderful, but I need the coordinates, please. Is there a snip track? No. No. Snip track? Snip track. Ugh. This is so much easier. Oh, thank you for that range, by the way. I do appreciate it, Maddie. I need to know where this is. Okay. Gene, near the C11 or 53. C11 or 53. C11 or 53. Well, that's frustrating. Okay. I hope this is something I can search. Yes. C11 or 50. It said near C11 or 53, unfortunately. Well, there's a snip near it. That's actually not helpful. Well, that's super disappointing. Sending me something. Maybe. It might be what I'm looking for. The problem, though, is that if on Snippedia the link is red, that means there's no page for it, which suggests that for some reason it's just not annotated. So if I click on one of these other ones and go to it, it will give... Oh, bad gateway. They should provide the coordinates. Yeah. So it provides the coordinates. Where are my coordinates? Oh, here we go. Like chromosome 4, position whatever. Identification of two maternal transmission ratio distortion loci in pedigrees of the Framingham heart study. So the problem is that these red ones, including less likely to sneeze after eating dark chocolate, is red. So there's no page, meaning that I bet on this track that you sent me, which I will remember because that will be interesting and useful. Oh yeah, all Snippedia pages were downloaded with their contact checked to remove pages that were auto-generated. Yeah, so thank you for these people who made this. But I don't think it's going to be annotated. Shoot. Well, that's super sad. Coordinates. I'll find it someday. In the meantime, though, there are quite a number of other ones. I think you're looking to me an article that I just saw. So yeah, they do say it's on chromosome 11. I don't see the coordinates, though. And I think this page does not work, although that's the same... I've been getting that error, so, Well, that's interesting. That's unfortunate. There's some of these other ones that I could probably look at, like hoarding. Hoarding sounds interesting. So I will search for hoarding, the hoarding allele. Do I have the hoarding allele? Do I have the hoarding allele? Oh wait, NCBI. Okay, hold on. Maybe I just need to search a different thing. Okay, let's check this one first. So it does not appear that I am a hoarder as long as I'm on. Is this HG19? Is that the same genome assembly? Oh, it's not. To HG19. You're not, it's not, you're unsupported. This is going to anger me a lot. Do I have to? I had to download them. Wait, which one is it? Okay. Take two. Hoarding. Go. Still no. Well, nope, still no. Still no. I'm not a hoarder. Genetically. I'm not genetically a hoarder. I want to find at least one. I know from something hangry. That's a good one. I know from something else that I've done that I definitely, obviously, have Neanderthal DNA. That's not surprising. I want to find which Neanderthal DNA, though. All right. Chromosome 15, but then I have to go to the liftover and put this in, and then I have to put space. Then I have to copy this. Then I have to put space and add 1. Submit. View my conversions. Open this stupid file. Open file. And the answer is no. What a process. Yeah. Well, it would be easier if I just, okay. If I would just align my genome to the correct assembly. By correct, I mean not HG19 for some reason. I don't know why Dante Labs decided to do that. It's not surprising, but it is a little annoying. Where's my fear of heights? Ooh, a table of lots of SNPs. This company seems a little not correct. Yeah, well. So for anyone who isn't in the know, oh, thank you. I got my whole genome sequenced. But they did it bad, as in they were like, it wasn't exactly what they had promised. And then when I asked about it, they're like, oh, we don't promise that. And then they mysteriously found the missing data. Ah, cool. Okay. So this is a table of a lot of SNPs available through GitHub. They aren't functional annotations. Okay, cool. This is super useful, although only, why are only 4,000 though? That's actually pretty low. Not that I think that, you know, we should have all of these annotated, but I think we have more than this annotated. I'm going to keep this open because I might check it out later, offline. Sense of direction, though. I gotta have a sense of, I must have a Neanderthal sense of direction. Where's my leftover? Why did I decide to type this? Yeah, so list of things I want to do with my genome. I want to get a better idea of the SNP content that I have, right? I also want to realign it to the T to T project genome, which is the telomere to telomere project, to get a more accurate, a different view. Nope. A different view of my DNA, a more complete view of my DNA. But I also want to put it into a genome that is more compatible, so I don't have to keep doing this back and forth thing. All right, I'm getting pretty sick, pretty tired of that. Won't just one of these work for me though? Why does it keep giving me? Why is it giving me a bad gateway? Snippedia just hates me right now. I also really want to turn my genome into something like, I don't know, like almost like an art project. But I've been doing some math, and there is practically no way to view, not just view, but okay, so I can make something called an imputed genome, where basically I look around, use bioinformatically, I look at all these reads and find all the weird stuff, like this right here. This is probably not real necessarily, but let's pretend this is a small deletion in my genome that doesn't exist in the reference genome. Well, I can modify a reference genome file to include that. And then I find a SNP, a small difference in my DNA that only I have, like this one. Not only I, but that I have, like this one. And I include that. Well, if I do that for everything, everywhere, I have what is my DNA, the thing, the actual DNA, not just the reference DNA. It includes all my SNPs and all that. And I was thinking, like, okay, that'd be kind of cool. What if I took that information and went down like here, this sequence track area. Show translation? The sequence track down here. See how they're different colors? Well, what if I make something pretty with that? Like, what if I printed them all out? Like the smallest dot I possibly can in these different colors, you know, front and back on a page. Like, how long would that be? Well, it turns out that book would be something like 100,000 pages long, even at the closest printing density that I can possibly get. So that's not going to happen, unfortunately. So I really want to figure out something. I also like calculated, like, what if I made like a little box, a little LED panel with colored lights that flipped through all my DNA, like just changed the color of the LED. I think I did my calculation. If it was like, depending on the size, it would take days to weeks to months to actually cycle through my entire genome. I don't know. I want something like that, but I'm A, not artistic enough, and B, not clever enough at this time to get around the problem that there's 3 billion bases to deal with. 3 billion if I'm doing the haploid genome, which I would just concatenate like this, the SNPs into, or heterozygous SNPs into like some other color or code. Like this, I would display the fact that it's a C and a T at the same time. The other idea is to be the first person to publish my genome on Amazon Books, which actually I should double check that. Publish genome on Amazon Books. What's it called? Amazon... Not genomics, not the genome defense. No, not this book, which I really should read. But literally, you know, a digital book, hundreds of thousands of pages long, just with my DNA. Probably anonymized in some way, but we'll see. All right. I do have to mention, so just to kind of go back. Ah. To Riker. This is just a whole new level of first. Yeah, I mean, I don't know. Like they have sorry, Riker, we're tangenting again. What is it called? Micro glass etch. Like you can get these little itty bitty things. I'm not finding it. This like data storage medium that's like etched glass, but really, really small. You could do an animated circos plot where you highlight X SNPs per linear chromosome and highlight them with different colors based on the SNP outcome. That is true. That is true. That's that might not be so bad. I don't know. I wanted a little bit more fidelity. You know what I mean? Like if you wanted to, you could go in and read what the bases are. Because obviously if I zoom out enough, yeah. Meanwhile, back to the Enterprise. Back to the Enterprise, and then it completely immediately off the Enterprise. Oh yeah. Maddie, I have my own rabbit hole emote. It's not animated though. I'm jealous of the animation. That's from Science, right? That looks so familiar. Oh yeah, absolutely is. Yeah, I wonder what proportion of Science streamers at some point think to themselves, I really need a rabbit hole emote. I bet it's the majority. I bet it's 100%. Sounds like time for a shiny app with multiple tabs. Oh, don't get me started on shiny apps. In that they're fun, but I'm really bad at making them, so they end up being awful. You use that one a lot. After your last stream, you need a rant emote. Would that be like a little phage, like yelling or something like that? I still want more emotes, more and more emotes. I have sort of good but bad news. That streaming that I did a couple of weeks ago, like very consistent streaming, put me ahead. I'm ahead of schedule on getting those free emote slots. Bad news is I saw in the fine print that the 40 streaming days, or no, 60 streaming days is after you've made affiliate, which didn't happen for me until like a month and a half to 2 months after I started streaming. So all those streams before then don't count. So I'm further behind than I thought. Yeah, I really want more emotes, not just in Twitch, but also in Discord. If it isn't too much to ask, if you have an idea for an emote, throw it on Discord so I can make it. I feel like I need more. I need more. I live off of them. What's the smallest storage medium? Single atom memory. That's a little too small. I want something readable. Micro etch glass. I'm remembering something from like Wired or Popular Science from back when I was a teenager. Like there was this proposal that you would store information on like micro etched glass or something like that. Yes, Brian. Thank you for clarifying. Larger than my DNA, because if we're looking for the small storage medium for my DNA, I got some right here. Perfect. Smaller bigger than a nucleus, smaller than a USB drive, or smaller than a 100,000 page book, double-sided, printed in color. I need your biggest bird feeder. No, that's too big. There's a Simpsons quote for everything. The irony here is it's probably going to be that DNA-based storage that they've been working on. You sequence it together. Ugh, yeah. You want a sassy tardigrade with a tongue sticking out for this situation. Throw it on Discord. I have a misc suggestions. Please throw it on Discord. I'll make it. In fact, why don't I make it now? Also, Maddie. Ugh. Okay. James, we're gonna talk about this bullshit when we talk to David about the quantum biology thing, because I noticed a part of the book, which I haven't gotten to yet, is about information storage. I am such a pessimist when it comes to storing useful information into DNA, right? We all know, not we all, many of us know that DNA tends to do things like recombine. It tends to mutate. The GC content can really fuck it all up. You also have the problem of like, oh, well, you just sequence it to get it back. Oh, okay. Maybe that's a little bit more possible now if you do enough nanopore sequencing or fragment it in a way that you can put together. But then you got to synthesize the thing. Someone tell me how we're going to synthesize all of Shakespeare's work. Also, Maddie, thank you for this article. I'm not mad at you. I'm mad at this concept. Ah, shit, I got some DNAs on my Kindle. my book library is gone. Yeah, we sweat enzymes that dissolve DNA and destroy DNA. How even so they did I'm gonna say it's George Church DNA GIF. Yeah. Haha. So okay. Well, that's real small. That's real small. So scientists used a very clever technique to store the image on the left into DNA on the right. Notice how it looks not good. Which is fine. First iteration, they did it absolutely. But the amount of space required to store something usable in terms of linear DNA, we're talking like chromosome length. Good luck getting a cell to deal with that. Good luck handling that without shearing the thing in a billion pieces every time you move it. Oh, and don't get it too hot or too cold. Or get too much water and then get too cold. Only if you stored in enough water or a proper buffer, then you're probably fine. Okay, wait. I do want to read this. Ugh. An unexpected confluence of furtive love. I'll try to use my ASMR voice. Although I've been talking a lot, so not good. In an unexpected confluence of furtive love, lust, and nucleotides, researchers have proposed a new method for stored data, transforming information, including poetry, into DNA. Only William Shakespeare could truly appreciate the scientist's choice of his own sonnets to demonstrate their high-tech prowess. It's a moment where some of the finest couplets and rhymes ever written literally merge with the basic chemistry of life, and 400-year-old lines of verse are translated into A's, C's, G's, and T's. I tripped up because I normally say A, T, C, G. Yeah, barf is right. This person pulled out their thesaurus. I feel like you can't talk about Shakespeare without pulling out your thesaurus. He didn't realize this was written not so great. Well, see, the problem is it was written in DNA and then translated for the Atlantic. It cost $100 billion, so they ran out of time to copy it or to edit it. Okay, blah, blah, blah. Okay, the research didn't merely transpose English letters into genetic code, they made real DNA. Okay, we'll look at if they have some paper, we'll look at it. Or some paper, a paper, some paper. Instead of coding for proteins, though, these info molecules code for say, the 2 lines of the famous Sonnet 18. Shall I compare thee to a summer's day? Thou art more lovely and more temperate. Transposed into DNA, a code devised by Nick Goldman and Ewan Burney of the European Bioinformatics Institute. The first 3 words of the sonnet. Oh, first 3 words? Okay, see, this is already the problem. Skimming it before, I thought they'd come up with a clever method to go from this to this. No, it's shall I compare is TAG, ATG, TGT, ACA, AGA, CTA, GC, whatever. As reported in Nature, each letter in the words "thou art more" is first converted into binary code of 1s and 0s, then translated through a process into 5 distinct DNA letters. For instance, the T in the word "thou" becomes okay, a 5 mer. Okay, wait. It's getting late. My brain's not working. And the vaccines are starting to make me feel more sickly, which I got today. Get your vaccines. How many combinations are there? So that's 4 times 4 times 4 times 4, right? 4 to the power of 5. Why did they do that? Okay. They dreamed up their project at a pub, jot down their thoughts on a napkin. Yeah. Later, they studied past efforts to use DNA. Yep. It's a really important milestone, says George Church. Thank you. No. So yeah, I wonder what the 1024, like, why did they use that many? They could save a lot of space. I'm sure. I'm just skimming now. I hope this Nature. Oh, good. It looks like a real address. It's not. That wasn't actually the paper. Why? I bet Sonnet goes. Yeah. Boo! Scared the shit out of me. Classic. We're drunk enough to think this nonsense, but sober enough to be able to follow through. Yeah. All right. I want this though. So we have Goldman and Burney. Goldman, Burney. DNA, Nature. Wait, this was Nature? Oops. What year was this? Ooh, 2013. Even worse. Be gone, horse. All right. Previous DNA-based information storage approaches have encoded only trivial amounts of information or were not amenable to scaling up and used no robust error correction. All right, that's something I agree with. Lacked examination of their cost efficiency. Here we describe a scalable method that can reliably store more information than has been handled before. We encoded computer files totaling 739 kilobytes of hard disk storage with an estimated Shannon information of 5.2 times 10 to the 6 bits into DNA code. Synthesize this code, sequence it, and reconstruct the original files. All right, great. I can't get it. I'm just going to take this information, go off screen here. Definitely not go somewhere with free articles. No, I got it. Ah. Where should I save this? Desktop. Yeah, do that thing. Here we go. All right. Paper time. Nick Goldman. All right. Also, since I was doing this a little bit on my own without having read this, I can kind of compare and contrast. So I see. Okay, interesting. So they took a binary text file and they re-encoded it into base 3 because it's 0, 1, 2 TAG, but see, these don't line up quite the same. So 2 does not mean T, 0 does not mean A. I'm sure there's some extra information. Okay, so they encode that into DNA, and then they have DNA encoded indexing information added. So they have alternate fragments have file information reverse complemented. Okay, so these double-stranded or single-stranded? So binary digits holding the ASCII codes for part of Shakespeare's Sonnet 18 was converted to base 3, read, using Huffman's code that replaces each byte with a 5 or 6 base 3 digit, or a trit. This in turn was converted to in silico to DNA code by replacement of each trit with one of 3 nucleotides different from a previous one used, ensuring no homopolymers. That is smart. You got to run? Thank you very much. Thanks for stopping by. I look forward to your next lab stream. Wait, you already got here. Well, you don't have to wait. You can leave. Did I do this right? There we go. Everyone should go check out James's channel. Someday, in the far-flung future, we're going to be talking with David Hewlett again, and us two scientists about quantum biology. Maybe, hopefully, if we can get our act together and schedule something. At least I'm reading the book. Trying to read the book. All right. Yeah, sometime. Okay, so they have no homopolymers, meaning that they don't have huge long tracks of the same sequence, which is important. So this formed the basis for a large number of overlapping segments of length 100 bases with overlap of 75, creating 4-fold redundancy. All right. Indexing DNA codes were added in yellow, also encoded as non-repeating DNA nucleotides. Okay. And then they did a bunch of scaling for costs. And then they show how it's less expensive. Oh, that's it. Well, that was pretty quick. Did they actually do this though? So we find that with current technology and our encoding scheme, DNA-based storage may be cost-effective for archival of several megabytes with a 600 to 5,000 year horizon. Excuse me? Does that mean... I'm a little confused. Does that mean they predict in 600 years it's more cost effective? I don't know, maybe. I understand that this error thing that they're interested in is important, but this is actually pretty old at this point. This is from 2013, and also is like has a printing error. 00 month 2013 volume 000. Interesting. Oh, that might be because of where I got it, maybe. I feel like there's a way to encode this a little bit more carefully. This goes back to my DNA programming thing, where I feel in my heart there's a way to do it that's better. This redundancy is a practical thing and kind of an interesting idea. But do you need that? Because so this is in like a pool. So this would be important if you had one version of every molecule. But you don't. You have millions, billions of versions of every molecule depending on how you make it and how you store it. I don't know. With negligible computational cost and optimized use of technologies we employed, we estimate the current cost to be $12,000 per megabyte for information storage in DNA and $200 per megabyte for information decoding. Modeling relative long-term costs of archiving using DNA-based storage or magnetic tape shows... Okay, so they're comparing it to magnetic tape, which is not fair. It's a very stable but very old technology that I feel like hasn't changed, but maybe someone who knows more about information technology science knows more than me. The key parameters are the ratio of one-time costs of synthesizing the DNA to the recurrent fixed costs of transferring DNA between tape technologies or media. Okay. So where did they get this information? This really is kind of like back of the... Could rewrite something like this? I mean, I'm curious about where they got these numbers, like which company they're talking to. What's IDT's full name? Integrated. Nope. I don't know. This is interesting though. I'm gonna read that more later. I bet we can do better. I bet on stream we could do better. All right. Thank you everyone for sending me things. I think that's going to be about it for me tonight. So thank you for coming. I'll be back on Sunday with microscope time. Yeah, I don't really have any announcements or anything like that. Other than there are some new chat point rewards, and if you resubscribe and make sure to add in some text, a little talking tardigrade should show up on screen, but we'll have to test that when someone actually does that. So lovely to lurk on while you write an article for your students about transcription and translation. Oh, that's great. I hope are you doing any activities for that? I feel like that's the way that I have found works like the best. I don't know, just having to like sit and write down and do transcription and translation yourself is such a good way to learn those processes, specifically those processes. I remember we have this activity that we did in grad school with children, which actually works still pretty well with older students, where you like encode or decode your name into DNA and RNA. They asked you to do more articles with vocab as previews after you did it today. They ask and you deliver. Wait, so they're asking for more information? They're asking for help? That's amazing. Absolutely amazing. Also, good for you for following through. I mean, I feel like we all know this, but I'm positive you're an amazing teacher. And I'm jealous for not having had you for my own 9th grade science education. They said that doing the vocab like that helps them understand the notes more when we go to write it down. Yeah, that's cool. That makes sense too. That's great. Wow. That's cool of your students to be like caring about their own education. That's an important step of like metacognition too, is identifying and understanding deficits. You made them all homologous chromosomes today. Nice. With alphabet alleles, and it was so fun. Oh, that's so cool. If I can find it and share, I know I was just about to like finish up, but whatever, I don't care. If I can find it, I was just reminded because someone is using my teaching material from last year. I made this thing that I just loved so much. I have a picture of a cat. Oh. Yeah, okay. Wait, let me find it. It's so dumb. Wow, literally metacognition. How strange. I found this. I gotta find it. Maybe it's not this. Maybe it's not this one. Oh, yes, here it is. Here it is. Where I was trying to help them understand. My students were very much struggling. My students had not really had very good biology instruction in high school, and so they were really struggling the difference between sister chromatids and homologous chromosomes. So we walked through an example, and this is the thing that stuck with them like all semester. Like every time I was like homologous chromosomes, they would be like, those are like two dogs that are that look like each other but aren't the same. It's like, yes, you understand homology. It was a small victories for my class last year. All right. You got red and blue fan sticks and number them randomly 1 to 23. And then put ABC/ABC or big ABC on the different chromosomes and made them go find their pairs. Oh, that's clever. I like that. That's cool. You made them write it down, and then they had to look at how they all had ABCs, alphabet genes, but different forms. That's cool. Or maybe the same forms. Yeah, that's cool. I like that a lot. So they actually had to get up and move, right? I assume that is totally something that is easier to get younger high school students to do rather than certain college courses. People just kind of want to sit down and listen. So you hope to get back to it when we get to heterozygous and homozygous. Yeah, that's smart. I wonder if you could. Oh, yes, they had to go find their pairs to corny music. Of course, corny music. I hope it was royalty-free corny music too, just to, add a little bit extra. I like that. I like also planting the seeds for the future. Doing stuff like that is always so smart. All right. Big cam. Big out of focus cam? Does it always look like that? Okay. Just go to big cam just so I can see my stuff more easily and so you can't see mine. So I hope everyone has a great night. We'll raid someone. But I'm not going to stick around. I have to get to bed. I'm starting to really feel this these vaccinations. Getting a little bit of a reaction. That's totally fine. Just means that my immune system works. We'll read Turtles and Chill. Super nice. All right. Good night, everybody. And I will see you next time. Stick around for the raid. All right. Have a good night. Bye.